Welcome to the SWAYAM course of Microbial Technology. Today we will be discussing about organization of prokaryotic and eukaryotic cell structure. Myself, Shilpi Banerjee, will be discussing about the different parts of prokaryotes and eukaryotes in terms of their cellular structure. Well, in accordance to this concept, previously you have seen that we dealt with what is microbiology, some of its historical concepts which is associated with microbiology, major contributions made by various scientists in the field of microbiology, what are the current trends going on in microbiology, scope and future of microbiology, and in last summary was discussed. In this lecture, we will be discussing about what is cell, characteristics of cell, types of cell, certain examples with pictorial representation, their organization, their structure, their functions and conclusion. Moving on to the introduction. Students, we all know what is cell, right? A cell is the structural and fundamental unit of life. The study of cells from its basic structure to the functions of every cell organelle is called cell biology. The first biologist who discovered cells in 1665 was Robert Hooke. Basically, all organisms are made up of cells. They may be made up of single cell, which is unicellular, or may be made up of many cells, which is basically multicellular organism. Mycoplasmas are the smallest known cells. Cells are the building blocks of all living beings. They provide structure to a body and convert the nutrients taken from the food into energy. Based on the organization of these cellular structures, all living cells can be divided into two groups, prokaryotic and eukaryotic. Animals, plants, fungi, protozoans and algae all possess eukaryotic cell types. Only bacteria has prokaryotic cell type. Prokaryotic cells are generally much smaller and more simple as compared to eukaryotic. Prokaryotes are in fact very much structurally simple because of their simple size. In all life forms, including bacteria, plants, animals and humans, the cell was defined as the most basic structural and functional unit. Based on scientific observations over the next 150 years and probably more, scientists formulated the cell theory, which is used for all living organisms no matter how simple or complex. The cell theory, which was given by Sheldon and Swan, incorporates three principles. Cells are the most basic units of life. Second, all living things are composed of cells. Third, new cells are made from pre-existing cells, which divides into two. As you can see in this pictorial representation, cell is the basic unit of life. The two cell which is showed in color graphics depicts eukaryotes and prokaryotes. The blue one shows eukaryote, it is more complex, there are many cell organelles. The green one depicts prokaryotes. At the simplest level of construction, all cells possess a few fundamental components. These include cytoplasm. Cytoplasm is what? A gel like substance, right? A jelly which is composed of water and dissolved chemicals needed for growth, and it is contained within a plasma membrane which is also known as cell membrane or cytoplasmic membrane. One or more chromosomes which also contains the genetic blueprints of a cell, and ribosomes organelles are used, which is basically used for production of proteins, right. Now, let us look on to the characteristics of cell. It can be any cell, prokaryote or eukaryote. So, cells basically provides structure and support to the body of an organism. The interior of a cell is organized into different individual organelles surrounded by a separate membrane. The nucleus, which is the major organelle in any cell, holds genetic information necessary for reproduction and cell growth. Every cell has one nucleus and membrane bound organelles in cytoplasm. Mitochondria, which is a double membrane bound organelle, is mainly responsible for energy transactions vital for the survival of a cell. Lysosomes are responsible for digesting unwanted materials in a cell. Endoplasmic reticulum plays a significant role in the internal organization of cell by synthesizing selective molecules and processing, directing and sorting them to their appropriate locations. Beyond these basic components which we have just now discussed, cells can vary greatly between organisms and even within the same multicellular organism. The two largest categories of cells which I mentioned earlier is prokaryotic and eukaryotic cell. They are defined by major differences in several cell structures. Prokaryotic cells lack a nucleus surrounded by a complex nuclear membrane and generally have a very simple circular chromosome located in a nucleoid. Eukaryotic cells have a nucleus surrounded by a complex nuclear membrane that contains multiple rod shaped chromosomes. All plant cells and animal cells are eukaryotic, even human cells are eukaryotic. Some microorganisms are composed of prokaryotic cells, whereas others are composed of eukaryotic cells. Prokaryotic microorganisms are classified within the domains of archaea and bacteria, whereas eukaryotic organisms are classified within the domain eukarya. The structures inside a cell are analogous to the organs inside a human body, with unique structures suited to specific function. Some of the structures found in prokaryotic cells are similar to those found in some eukaryotic cells, others are unique to prokaryotes. Although there are some exceptions, eukaryotic cells tend to be larger than prokaryotic cells. This comparatively larger cell of eukaryotic cell dictates the need to compartmentalize various chemical processes within different areas of cell using complex membrane bound organelles. In contrast, as you can see in the pictorial representation of both the cells, prokaryotic cells generally lack membrane bound organelles. However, they often contain inclusions that compartmentalize their cytoplasm. Now, let us look on to the details of the main two types of cell, prokaryotes and eukaryotes. Pro is what, students? Pro means primitive and karyon means nucleus. This means that prokaryotic cells are those which contains primitive nucleus. The true nucleus is absent in case of prokaryotes. The primitive nucleus is also known as nucleoid. It is just a region where the DNA of a cell is present. The cell wall of prokaryotes is non-cellulosic and very complex in nature. Their size usually ranges from 3 to 5 micrometer. Their cell cycle is not divided into phases. Membrane bound organelles like endoplasmic reticulum, Golgi apparatus, plastid, mitochondria, centriole, vacuole, vesicles are absent. Cilia is also absent in prokaryotic cells. Flagella are made up of flagellin protein which are randomly arranged and it has 3 parts, namely basal body, hook and filament. Some specialized structures like pili, fimbriae, mesosomes, plasmid and inclusion bodies are only present in prokaryotic cells. Examples of prokaryote includes the whole Monera kingdom. Now, let us look into the details of organization and structure of prokaryotes. As you can see, a prokaryotic cell has flagellum, pili, capsule, cell wall, plasma membrane, nucleoid, cytoplasma and ribosome. Now, what is capsule? This is present in some bacterial cells, which basically is an outer covering and it protects the entire cell when it is engulfed by other organisms, which assist in retaining moisture and helps the cell to adhere to surfaces and nutrients. The next important structural part is cell wall. The cell wall is an outer covering that protects the bacterial cell and gives it shape. Cytoplasm is a gel-like substance which has been mentioned earlier, which is composed mainly of water and also contains enzymes, salts, cell components and various organic molecules. The cell membrane, also known as plasma membrane, surrounds the cell cytoplasm and regulates the flow of substances in and outside the cell. Pili. It is basically a hair-like structure present on the surface of a cell that helps to attach to other bacterial cells. Shorter pili is known as fimbriae, help bacteria to attach to surfaces. Flagella is long, whip-like protrusions which helps in locomotion of prokaryotes or certain bacteria. Ribosomes are protein machinery, they are cell structures responsible for production of protein. Now, what is plasmid? Plasmids are gene carrying circular DNA structure which is not involved in reproduction process. Now, what is nucleoid region? So, area of cytoplasm that contains the single bacterial DNA molecule is nucleoid region. Prokaryotic cell lack cell organelles which is present in eukaryotic cell such as mitochondria, endoplasmic reticulum, Golgi apparatus and many others. Like plant cells, bacteria also have a cell wall, we just now saw, right. So, some bacteria also have a additional polysaccharide capsule layer surrounding the cell wall. This is the layer where bacteria produce biofilm, a slimy substance that helps bacterial colonies adhere to surfaces and each other for protection against antibiotics, chemicals and other hazardous substances. Similar to plants and algae, some prokaryotes also have photosynthetic pigments. These light absorbing pigments enable photosynthetic bacteria to obtain nutrition from light. Now, let us look into the organization and structure of eukaryotes. The term eukaryote is derived from Greek word. Eu means true and karyon means nucleus, right. Karyon we just now saw in the case of prokaryotes, right. So, that means what? True nucleus. So, prokaryote was what? Pro-nucleus, primitive nucleus. Eukaryote is what? True nucleus. So, eukaryotic cells are defined as cells that contain a true nucleus or organized nucleus and many membrane bound organelles which is important for its survival. They have a more advanced structural organization that is large and more complex than a prokaryotic cell. However, they share a common features including the cytoplasm. The first eukaryotic cell probably evolved about some 2 billion years ago. The endosymbiotic theory explains the evolution. So, according to the endosymbiotic theory, large cells engulfed small cells, but are not digested by them. Instead, the small cells lived within them and evolved into different organelles. The large and small cells formed a symbiotic relationship where both cells benefited from each other, hence the name endosymbiosis, right. So, endosymbiotic theory is a theory suggesting that the organelle such as let us say mitochondria and chloroplast within eukaryotic cell came about as a result of early endosymbiosis between prokaryotic endosymbionts and eukaryotic host cell. This form of symbiosis involves a larger cell that serves as a host and a smaller cell that is referred to as endosymbiont. The larger cell engulfed or took in the smaller cell. The larger cell represents the eukaryotic cell of today and smaller cell represents the prokaryotic cell. This has been explained in the pictorial representation. Now, eukaryotic cells are cells that contain a membrane bound nucleus, a structural feature which is not present in any other bacterial or archaeal cells. In addition to this, in addition to the nucleus, eukaryotic cells are characterized by numerous membrane bound nucleus such as endoplasmic reticulum, Golgi apparatus, chloroplast, mitochondria and others. Now, let us look on to the plasma membrane. Like bacteria and archaea, eukaryotic cells have a plasma membrane. It is basically like a sandwich. Now, how? Let us see the details. It is basically a phospholipid bilayer with embedded proteins that separates the internal contents of cell from its surrounding environment. The plasma membrane controls the passage of organic molecules, ions, water and oxygen in and out of the cell. Waste such as carbon dioxide and ammonia also leave the cell by passing through the plasma membrane, usually done with taking help of some protein transporters. The plasma membrane is a semi-permeable membrane that separates the inside of a cell from outside. In eukaryotic cell, the plasma membrane consists of protein, carbohydrate and two layers of phospholipid, that is lipid having a phosphate group. So, these phospholipid are arranged in two ways. The polar hydrophilic. Now, what is hydrophilic? Water loving. This polar hydrophilic head face the outside and inside of cell. These head interact with the aqueous environment outside and within a cell. Now, the second layer, that is the non-polar and hydrophobic, which is water hating or water repelling, tails are sandwiched between the heads and are protected from the aqueous environment. Now, this entire concept, this entire structure of the plasma membrane was given by two scientists, that is Singer and Nicholson, who described the structure of phospholipid as fluid mosaic model. Why? Because this bilayer looks like a mosaic, right, a kind of brick structure and has a semi-fluid nature that allows lateral movement of proteins within the bilayer. Now, what is the function? So, since it is a membrane, it is obvious that movement of certain selected substances will be allowed. So, the plasma membrane is selectively permeable, that is, it allows only selected substances to pass through. It protects the cells from shocks and injuries. The fluid nature of the membrane allows the interaction of molecules within the membrane. It is also important for secretion, cell growth and division. It allows transport of molecules across the membrane. The transport can be done in two forms: active transport, passive transport. Now, what is active transport? So, this transport occurs against the concentration gradient and therefore requires energy in form of ATP. It also needs carrier proteins and is highly selective process. Passive transport occurs along the concentration gradient and therefore does not require energy. Thus, it does not need carrier proteins and is not selective. Now, let us look on to the cell wall. Cell wall is a non-living rigid structure outside the plasma membrane in plant cells and fungi. It is absent in eukaryotic cells of animals. The structure and composition of cell wall is made up of different components in different eukaryotes. Cellulose, hemicellulose, protein and pectin is present in plants. Cellulose, gelatin, mannan and calcium carbonate is present in fungi. So, this is the reason I mentioned that structure and composition of cell wall is made up of different components in different eukaryotes. Okay. The cell wall is divided into different layers. The middle lamella is the outermost layer and is made up of calcium pectate, which helps in adjoining the cells together. Next is primary layer. It is the middle layer and is made up of cellulose and hemicellulose. It is present in young growing cells and is capable of growth. Next is secondary wall. It is the innermost layer and is similar in composition to the primary wall. That means, this also contains cellulose and hemicellulose. What is the function? So, wall itself tells you is for providing shape or protecting something from injury, right. So, cell wall means it provides shape and structure to the cell, it helps in interaction between cell to cell, and of course, it protects the cell from injury or any undesirable molecules or entry of pathogens. Okay. So, moving on to the next structural part of eukaryotes, that is cytoplasm, which is common in both prokaryotes and eukaryotes. So, cytoplasm refers to the entire region of a cell between the plasma membrane and nuclear envelope. You can see in this pictorial representation. It is composed of organelles suspended in a gel like cytosol, cytoskeleton and various chemicals. Even though the cytoplasm consists of 70 to 80% water, it is nevertheless, it has a semi-solid consistency. Now, let us look into what is nucleus. Typically, nucleus is the boss of any cell. Nucleus is the most prominent organelle in a cell when viewed through a microscope. It houses the cell's DNA. It is the largest organelle of a eukaryotic cell, accounting for around 10% of its volume. It houses the genome, and through gene expression, it coordinates the activities of a cell. The nucleus itself is comprised of distinct components, and understanding their structure allows a deeper understanding of their function. Nucleus composes of nuclear envelope. So, what is nuclear envelope? It is a structure that constitutes the outermost boundary of nucleus, which is a double membrane. Both the inner and outer membranes of nuclear envelope are phospholipid bilayers. The nuclear envelope is also punctuated with protein based pores that control the passage of ions, molecules and RNA between the nucleoplasm and cytoplasm. The nucleoplasm is the semi-solid fluid inside the nucleus, where we find the chromatin and the nucleolus, a condensed region of chromatin where ribosome synthesis occurs. Now, let us look into what is chromatin and chromosome. So, chromosomes are thread-like structures within the nucleus that are made up of DNA and is basically the hereditary material which is passed on from one generation to another generation. In eukaryotes, chromosomes are linear structures. Every eukaryotic species has a specific number of chromosomes in the nuclei of its cell. In humans, for example, the chromosome number is 23, whereas in fruit fly it is 4. So, it is different for every organism. Chromosomes are only clearly visible and distinguishable from one another by visible optical microscopy when the cell is preparing to divide, and the DNA is tightly packed by proteins into easily distinguishable shapes. When the cell is in the growth and maintenance phases of its life cycle, numerous proteins are still associated with nucleic acid, but the DNA strands more closely resemble an unwound jumbled bunch of threads. The term chromatin is used to describe chromosome when they are both condensed and decondensed. Now, let us look into what is the nucleus, nucleolus. Some chromosomes have sections of DNA that encode ribosomal RNA. A darkly staining area within the nucleus called nucleolus aggregates the ribosomal RNA with associated proteins to assemble the ribosomal subunits, which is then transported out to the cytoplasm through the pores in the nuclear envelope. Nuclear lamina provides mechanical support to the nucleus. This is a protein mesh which is more organized on the internal surface of the nucleus than on the outer surface. Now, let us say what is Golgi apparatus. So, it is basically made up of many flat disc shaped structures called cisternae. It is found in every eukaryotic cell except human red blood cell and sieve cells of certain plants. The cisternae are arranged concentrically in parallel to the nucleus. It has a cis face that faces the cell membrane and the trans face that faces the nucleus. Functions of Golgi apparatus is basically acts as a packaging material within the cell. It helps in modifying and maturing the proteins. It also helps in production of glycoprotein and glycolipid. Now, let us look into what is endoplasmic reticulum. It is a small network of tubular structures. It divides the cell cytoplasm in two parts, luminal and cytoplasm. They are of two types, smooth endoplasmic reticulum and rough endoplasmic reticulum. Smooth means they do not have ribosome, whereas rough indicates ribosome is present. Okay. So, what is the function of endoplasmic reticulum? They help in lipid synthesis. They also carry out certain protein folding and transporting them to Golgi apparatus as a packaging material. They are also helpful in synthesis of protein. Now, let us move on to the mitochondria. So, mitochondria is nothing but powerhouse of a cell. All energy based molecules are produced here. It is a oval shaped structure which is bounded by two membranes. The outer and inner membrane divides the mitochondrial lumen into two compartments. The outer membrane surrounds the organelle while the inner membrane is semi permeable that forms folds called cristae. The region within the inner membrane is called the matrix and between the two membranes is called the intermembrane space. Mitochondria contains DNA, RNA and other components which is required for the synthesis of protein. Function, as I mentioned earlier, it is very important for producing energy in form of ATP, thus called powerhouse or energy currency of cell. It helps in regulating cell metabolism and it is also important for protein synthesis. Now, let us look on to the lysosomes. Membrane bound organelles formed in the Golgi apparatus basically represents lysosome. They contain rich hydrolytic enzymes such as lipase, protease and peptidase. Lysosomes are basically also known as suicidal bags. They help in digesting lipids, proteins, carbohydrates and nucleic acid. They also help in preventing the entry of foreign particles such as bacteria and virus and destroy them once they enter the cell through the process of phagocytosis. Now, let us look into peroxisomes. They are single membrane bound small round shaped structures. They contain digestive and oxidative enzymes. Peroxisomes are a group of heterogeneous organelles and the presence of marker enzyme distinguishes them from others. They help in detoxifying poisons or removing poisons inside the body through various oxidation reaction. They help in facilitating in photosynthesis and seed germination. They also help in converting stored fats into sugar, especially in case of plants. They help in metabolism, pathogen defense and stress response as well. Again, this is more suitable for plants. Into what is plastid. So, they are basically double membrane bound organelle found only in plants. Based on the type of pigment present, they are of three types: chloroplast, which contains green pigment, chromoplast, which contains green carotene, and leucoplast with no pigment at all. So, their function is to perform photosynthesis. It imparts flowers and fruits its yellow, red or orange color. It also helps in storing carbohydrates, oils and fats as well as proteins. Now, let us look into the vacuoles and vesicles. Vacuoles are found centrally in plants, making up almost 30 to 80% of the total plant cell volume. It is the largest organelle in plant cell, filled with fluid, ions, enzymes and other molecules. In animal cells, they are comparatively much smaller in size as if you compare with plant cell. So, vesicles are membrane bound sacs that can fuse with cell membrane or any other membrane system within cell. So, basically the function of vacuole and vesicle is to maintain the turgidity of cell, transporting ions and molecules in and outside the cell through the process of endocytosis or exocytosis. It also helps in storing reserve food, water and waste. Centrosome, which is present only in animal cell, is a microtubule organizing center. It contains a pair of centrioles that lie perpendicular to each other, which is clearly seen in this pictorial representation. Each centriole is cylindrical and comprised of 9 microtubule triplets. Their main function is in cell division. On a concluding note, the structure of both prokaryotic and eukaryotic cells underpins their respective abilities to perform essential functions, which adapts to environment and contribute to advancement in field from biotechnology to medicine. So, understanding these structures in detail enhances our knowledge of life, process and our ability to harness them for various application and research endeavors. In the next lecture of microbial technology, we will be dealing with microbial diversity. How vast is the world of microbes? What is their role? What are the examples associated? What are the different types? Bacteria, virus, protozoa, fungi, algae, helminths and many more. How is it beneficial to the society? How some of them are beneficial, how some of them are dangerous or act as pathogen, this all will be dealt in the next topic. Thank you very much. I hope we have been able to make you understand what is cell, their types, in terms of organization structure, their parts and how they are different from each other. Thank you very much.